Renzo Huber, who will talk about non-bold contrast to improve the interpretability of high-resolution fMRI in humans.
Thank you very much. And I also want to start by thanking Amir for inviting me. It's really a pleasure to be here and fun. And he asked me to talk about non-bold layer-dependent fMRI contrasts. So I will talk about non-bold layer-dependent fMRI methods, and I will list a few of them, the most popular ones, and briefly describe how they work. And then in the second half, I also want to talk about my favorite applications of non-bold layer-dependent fMRI, mostly with one very specific version. And this is an educational lecture, so I will not have the time to go very deep into the details. If you care about the details, I would like to recommend you this review article. And just in case you're the kind of person that doesn't want to read a lot of text and prefers to have things explained to you with pretty images, check out this YouTube playlist that is called Non-Bold Layer-Dependent fMRI, where I collected a lot of lectures from other experts in the field where they talk about their view of non-bold layer-dependent fMRI. And also, since this is only a 30-minute lecture, I want to apologize already if I cannot really mention all important contributions of non-bold layer-dependent fMRI in the field. So if you're in the audience and I will not show your contribution, please forgive me. This is specifically thinking about the fact that almost everything we know about non-bold laminar fMRI comes from decades of careful animal research. But today I will mostly focus on the human side of non-bold layer-dependent fMRI. And we already heard a bit about it already, and the reason why layers are so emerging and so nice is because they can help us to bridge different spatial regimes of neuroscience, all the way from the microscopic side, where we have synapses and neurons and clusters of neurons, up to the macroscopic side, where we have behavior, brain areas, and distributions within those brain areas. And the keystone here to bridge these spatial regimes will be the regime of a few hundred micrometers where we have these layers. And the kind of new information that we get about these layers refers to directional connectivity. For example, we know that in a hierarchically organized system, we have mostly feed-forward input in the middle layers and feedback input from other areas that are higher in the hierarchy, mostly in upper and or deeper layers. And there are also these other microcircuits around. For example, especially in the frontal areas, there's this kind of input-output model that I will also talk about. So layers tell us about something about the connectivity and where activity is coming from. To measure it, however, we already learned about this from Witzke, that it's complicated. We are not really measuring the neural activity directly, but we are measuring these vascular responses, and we are relying on the so-called neurovascular coupling, schematically depicted here as like firing neurons. We have an astrocyte connected to an arterial vessel, and there's also going a vein through here. Now, when we have activity increase, especially on the postsynaptic side, we have a lot of energy demand, and the arterial vessels are triggered to like open up, which means that we have an increase in blood volume, and the flow also shoots up, and a short while later, also on the venous side, we have these oxygenation changes. And you already see the two major limitations of layer-dependent fMRI here, namely, the first is that we are not quantitative. If we have like increased signal changes for one person or compared to another, or in one brain area compared to the other, we don't really know if this is due to different neural activities or if this is just due to all these different mechanisms that are involved here. The second big limitation is that the vein here, it comes from somewhere, right? It comes from somewhere over there, meaning that its oxygenation changes are not only reflecting the neural activity very close by in this layer, but might actually be coming from somewhere else. And this is summarized again here, suggesting that we care about the tissue activity, but we actually end up with this whole cascade of different mechanisms where the tissue activity induces blood volume change in the arterioles. We have oxygen consumption, all kinds of fluid dynamics, until we get to an oxygen oversupply that triggers a susceptibility-induced T2 star signal increase. And this thing at the end here is what we measure, while this thing at the beginning is what we care about. So it would be beneficial to literally get closer to the tissue activity and measure one of those first parameters directly here with non-bold layer-dependent fMRI, such as the vasodilation or the oxygen consumption, which means that we get rid of a lot of these unknown mechanisms down there. The second big limitation, as I already alluded you to, is that we have this spatial localization limitation in the sense that just increasing the resolution does not always help so much. Going from 3 millimeter to 2 millimeter, 1 or 0.7 millimeter means that we usually still end up with some kind of fuzzy blobs. And now we have these blobs at high resolution, but they're still kind of fuzzy and don't give us these stripy patterns that we would like to have with layer-dependent fMRI. And we already heard about this, that this mostly comes from the fact that BOLD is mostly sensitive to these oxygenation changes in the larger veins that are not as nicely laminar aligned like the neurons are. And there are other methods out there that are proposed to be more specific, such as these non-BOLD layer-dependent fMRI methods. And one of my favorite methods is this Vaser method, which stands for vascular space occupancy, which is believed to measure or be mostly sensitive to blood volume changes in the small vessels that are closely aligned to the laminar neural structures. And how this works is depicted on this slide. By assuming a two-compartment model. So I'm showing you two magnetization vectors here, a red one representing the blood magnetization and a blue one representing the tissue. And then we start every TR with a global 180-degree inversion pulse that flips around this magnetization, and then these two magnetization vectors decay with different T1 relaxation times. And there's one exact time point where the red arrow here is very small, is 0, and this is the time when we play out the excitation pulse and acquire our signal. So right during this exact time, during our inversion recovery curve, we are actually only sensitive to everything but the blood. So we don't have any red arrow available here. All the signal that we have available is the blue extravascular tissue. This means that for any voxel that might have all kind of different volume components, we actually see everything but the blood. So blood is black, does not contribute to the signal, and we only see everything around it. Now when we have activity increase, all the vessels open up, means that we have more of this black blood in our voxel, meaning that the signal goes down. And the signal decrease is then believed to be directly proportional to the blood volume increase. But there are also other non-BOLD layer-dependent fMRI methods out there. And another one that I want to talk about is called arterial spin labeling, ASL, in order to measure cerebral blood flow, CBF. And in these kind of measures, we don't really care about the amount of blood volume change you have within your voxel, but we care about the amount of blood volume delivery to the voxel along the feeding arteries. So for example, when we care about a slice group like here, we care only about how much blood is delivered, for example, from an area down there to our imaging planes. And this can be done by using this inversion pulse that I showed you on the previous slides in a specific location only, for example, only in the green areas down there. And then a short while later, we measure how much of this flipped magnetization arrives in our imaging plane. And the blood flow is then quantified as the difference of the signal with and without this application of this spin inversion down there. And to show you some real-life examples, I'm flipping back here between BOLD and CBF. And you can see that in CBF we have like less significant voxels, which could mean two things, right? It could mean that it's just more specific, right? That we have a reduced rate of false positives compared to BOLD. It could also mean that it's just less sensitive and it has an increased rate of false negatives, right? And in reality, it's usually a mixture of those. And if you are willing to scan for a long, long time, up to double-digit number of hours, then you can go down to like 0.8 millimeter isotropic resolutions and get pretty decent layer profiles of CBF activation changes down there. Yet another non-BOLD layer-dependent fMRI methodology is called calibrated BOLD, which is believed to give us estimates of the cerebral metabolic rate of oxygen, CMRO2, basically the amount of oxygen consumption. And it works as follows. So we know that the BOLD signal can be described as a function of all kinds of physiological parameters, most importantly the oxygen consumption, and also the vascular response here indicated as CBV. And we can try to tease apart these two independent physiological parameters with a series of different experiments. For example, in a first experiment, a hypercapnia calibration experiment, you give participants gas mixtures to breathe with increased concentrations of CO2, which is believed to only modulate this term here. All the vessels, they open up when they have more CO2 around without really affecting the oxygen consumption so much. And then in a second experiment, your neuroscientific experiment, you do any kind of neuroscience task, for example, a finger tapping, which does engage the neurons and also in response then engage the vasculature. And then we can try to budget what kind of signal contributions we have for these different experiments. So for example, for the gas calibration experiment, we do have vessel dilation, but no oxygen consumption. Whereas for the neuroscience tasks, we do have both. We have vessel dilation and oxygen consumption. So when you contrast those two experiments, look at the difference in this kind of equation, then we can tease apart the oxygen consumption. And how this looks like in real-life data is depicted here for a finger tapping task in the motor cortex, where you can see that for tapping activity, you mostly see activity in the hand knob for blood volume increase and also the BOLD, whereas for the hypercapnia gas calibration, you see that almost everything opens up. It basically gives us all the vessels open up, independent where you are, and it gives us some sort of vascular reactivity. And combining all these four different estimates here into this equation can give us estimates of the cerebral metabolic rate of oxygen across cortical depths too. And it's still kind of under investigation and debated how well these model assumptions really hold across a sub-millimeter layer-dependent regime. So I showed you a few non-BOLD layer-dependent fMRI methods already, and I think it's time to compare it with the gold standard, the gradient echo BOLD. And the most relevant quality features of any fMRI sequence that I could think of are the sensitivity, basically how well you can capture your signal, how long you need to scan, how strong does your task need to be, or can you also capture small signal changes, how specific you are to individual layers, how invasive you are, how available the sequence is, in the sense that do you need to be special friends with the vendors or have collaborators that help you with the programming, or how much effort in generally you need to put into getting data in the first place, how effective your sampling is and how quantifiable it is in terms of can you compare the activity across two different brain areas, across two different people. And for gradient echo BOLD, we have a lot of signal change, especially from the large vessels indicated here as different oxygenation values in the different color schemes here between activity and rest, meaning that BOLD, gradient echo BOLD, is super sensitive, but it's not really specific because a lot of this activity comes from layer unspecific veins. It's as non-invasive as it gets with MRI. It's super available on every scanner. The sampling efficiency is perfect. Its quantifiability is pretty bad, however. So we only get these percent signal changes, but we don't really know percent of what, right? And if it's bigger in another person or in another area, we don't really know where this might be coming from. So for ASL, we are not really sensitive to these large draining veins. They're kind of grayed out, and we are believed to be mostly sensitive to the water exchange of the capillary bed in the background, meaning that we have a much higher layer specificity. We are still non-invasive. The availability is getting pretty good, especially now that the patent ran out. The sampling efficiency is not the best, especially because you need to have these control and label images, but the quantifiability is higher in the sense that we can assign a meaningful physical unit to our results, milliliter per tissue volume per minute, for example. Now, for calibrated BOLD, to estimate CMR2, we are not really sensitive to the vessels in the first place, or at least we believe that. We are mostly sensitive to the extravascular oxygenation consumption changes indicated with different gray values here. And this method really shines on the quantifiability. We get a meaningful energy demand measure out of it, whereas all the other quality features are pretty bad. It's not even really non-invasive anymore because you need to give your participants these gas mixtures to breathe, which can be pretty uncomfortable. And also you need to have extra hardware available to deliver these gas mixtures, to have safety control mechanisms in place, so you're not really changing the concentrations too badly. And the last method that I talked about also is this Vaser method, which is not really the winner of any of those quality features, but it's actually also not the worst on any of those. So it gives us a good compromise to work with. It's not only specific to the very small vessels. In fact, there are also these smaller arterial vessels that are not completely layer specific that also contribute to the contrast. So in conclusion, for all these different methods, I think that all non-BOLD layer-dependent fMRI methods are noisier than the BOLD, that's for sure, and it's not just a question of how much of this reduced sensitivity is actually wanted reduced sensitivity, because we are not sensitive to these unwanted large vessels. all non-BOLD layer-dependent fMRI methods seem to be more specific and also more quantitative. And I talked about a lot of non-BOLD layer-dependent fMRI methods already, but there are actually many, many more, almost impossible to list. There's always like a new paper that you find where people did something different. And in fact, I only focused on the vascular-based non-BOLD layer-dependent fMRI methods, but there are also non-vascular methods. And maybe some of the most promising methods that are non-vascular might be here functional diffusion-weighted fMRI, where you can see my mouse is gone. Diffusion-weighted changes where you can see that with this method on the top right, you see that you can identify the middle layers pretty well without these venous biases towards the cortical surface. And the button seems to work still. Also, it has been shown that with T1 row sensitive fMRI, there are non-vascular changes of CSF volume, especially above the superficial layers, as visible in the bottom row here. So I think I have about 50 minutes left or so, so I would like to not only talk about the methods of non-BOLD layer-dependent fMRI, but also about some applications of non-BOLD layer-dependent fMRI, specifically for neuroscience applications. And the examples that I will show mostly refer to applications done at NIH in the group of Peter Banditini, also some groups together with Emily Finn. And the kind of beginning, how we started to do non-BOLD layer-dependent fMRI was in the motor cortex, where we already know what to expect from the layer-dependent connectivity pattern from animal tracer studies. Namely, we know that in the superficial layers, we have this cortical input, whereas in the deeper layers on the right, we have the cortical spinal output. And we try to engage these different inputs and outputs differently with different touching and moving tasks. And I would really like to have my mouse. Is it possible to get the mouse back? Like, I can click, but I cannot move it. And the light is gone, so maybe it's not attached properly. Yes, I got it, I think. The light is gone again. No. Sorry. There we are. I can try with clicking. Sorry about that. So we try to engage these upper and deeper layers differently with different tapping and moving tasks. So for the tapping, on the bottom left, you move your muscles, meaning that we do have some cortical spinal output, which is visible with the right loop on the right side. But we also have this touch component, meaning that there's a lot of cortical input, for example, feedback from sensory cortex or the motor planning area. So we also have a superficial activity, which you can see in the left plot here within the green highlighted areas, and also visible as the two peaks in the blue curve in the layer-dependent profiles. Now, for another task condition, for example, for the touching that is framed in black here, we ask the participants not to move their fingers, which does not result in activity of the deeper cortical spinal output layers. In fact, you see in the black framed middle activation map that the deeper layers are actually blue. It seems to be inhibited. Also visible in the black line in the bottom graph, you see there's only activity in the superficial layers, but nothing going on in the deeper layers. And this made us quite happy in the sense that we can differentially look at these different input and output circuits with different tasks. And it's not just scaled versions from each other. However, an important lesson that we learned from that is also that you cannot really do layer-dependent fMRI without knowing where you are in the columnar domain too. So for example, if I move this green highlighted area just a few millimeters away, you can see that the entire nice, interesting layer dissociation completely dies away. So we really need to know where we are in order to have good interpretation of layer-dependent signals. So we try to also develop this kind of topographical representations across layers and columns directly. Here, for example, looking at very small body part representations in the motor cortex. And to show you how this works in real-life data, I'm showing you this little animation here where I'm starting with an axial-like view of the central sulcus. On the left side would be the sensory cortex, where the scrolling algorithm now grows directly within voxel space, in black for the sensory cortex and in white for the motor cortex. And having these kind of voxel surfaces directly in EPI space helps us to look at the slices and the surfaces directly in the same space, also in 3D here, where I'm now basically rotating the entire central sulcus to look at it from the back of the brain. So on the left side would be the midline of the brain, on the right side would be where the mouse is, would be the right ear, and I can project the individual fingers onto it. So red is pinky finger, ring finger, middle finger, index finger, and thumb. And you can see that in the sensory cortex, they are nicely somatotopically aligned. They kind of span across the depth of the central sulcus, and it's pretty huge. It's about 30% of the entire central sulcus on the sensory side seems to be busy with finger representations. More interesting, however, is then what happens on the motor side. So when we switch this around and look at it now from the front perspective, and you see in the center this little cave, this would be the hand knob, and again I'm projecting the individual fingers onto it. Pinky finger, 2 ring fingers, 2 middle fingers, 2 index fingers in blue, and 2 thumbs in pink. And you can see that in the motor cortex, it's kind of more messed up. It's much, much smaller, and we have multiple kind of mirrored representations. And especially the right representation is as small as about like 4 millimeters, which might be better visible. I would like to advance my slides. Could you press the right key for me, please? The right key, the right arrow key. That's the one, thank you. So let's project it onto this plane, and then we can look at the motor cortex and the sensory cortex in the same view, where on the bottom left you see the huge finger representations of the sensory cortex and the smaller mirrored representations on the top right for the sensory cortex, for the motor cortex, sorry. I will repeat. On the bottom left, the sensory cortex. On the top right, the small ones are the motor cortex. And on the left, you see that with non-BOLD layer-dependent fMRI methods, these kind of mirrored representations are much easier to see compared to the BOLD, which also shows like indications of these mirrored representations, but as soon as you have one medium-sized vein around it somewhere, the kind of 2 millimeters around it are dominated by that finger. And these kind of representations are also highly consistent across participants here. And we also see that the patches that refer to entire mirrored finger representations also nicely match with patches in the motor cortex in the bottom line that preferentially are activated for grasping versus retraction movements. So the motor cortex at this small spatial regime is not only a body part representation map, but it's more an action representation map, as has been suggested from the animal literature too. So everything I showed you until now referred to the motor cortex, which is super easy. And people told me that like trying to understand how the brain works by looking at fMRI blobs is like trying to understand how a truck engine works by looking at it with an infrared camera. We only roughly know what's heating up and where energy is being used, but we don't know anything about the content, where the truck is going, or if this heat is coming from a densely packed microcircuitry, for example, from input or from output. Similarly, for fMRI in the motor cortex, we don't really know much about the content aside of the fact that the body part seems to be moving somehow. With layer-dependent fMRI, we are approaching this regime where we can say something about the densely packed microcircuitry, for example, counting the stripes down here where we have 2 stripes for tapping or 1 stripe for touching. But the motor cortex is so easy. It's not like any regular truck engine, but it's like the biggest engine there is. There's no cortex as thick as the motor cortex, and you don't need any fancy methodology to easily see the densely packed microcircuitry of an engine like that. And even with a small field of view, you can see this input and output activity. So we invested quite a bit in the last 3 years to extend this layer-dependent fMRI methodology to also work on more regular-sized cortices, such as the visual system. And we want to get there without really sacrificing this stripiness. And we actually still want to see these like dark voxels forming a line that I cannot point to now, indicating the stria of Gennari. And we want to have this not only in a small field of view, but actually in an entire slice, part of an entire slab of isotropic voxels. And since we care about activity, not only pretty EPIs, we need more than just the blobby BOLD, but also the higher specificity of Vaser, even though it comes along with a higher noise level too. And to see how applicable this is, we followed Jon Polimeni's approach, the next speaker, by looking at resting state connectivity patterns between LGN, V1, and V5, which have different feed-forward and feedback connections. And visible here, for example, for functional connectivity, resting state correlations across cortical depth within V1, you see that we have a clear middle feed-forward peak for a seed region in thalamus, where we also have a feedback peak for feedback input from V5. And this made us, convinced us that we can use this non-BOLD layer-dependent fMRI also on thinner cortices, and we tried to move beyond that and also apply these kind of layer-dependent non-BOLD fMRI methods not only in validation experiments, but also move beyond these very well-controlled cases where we zoom into a patch of cortex and then look at differential responses for different seed regions or different task conditions. Why not do it the other way around and look at one task condition, but look at differential responses across the columnar domain? So again, here we are assuming a canonical microcircuit, and we just assume that a feed-forward-driven column would look like the red one down there, whereas a feedback-driven column would look like the blue one down there. And then we can use any resting state data, pick a seed region, and cluster the brain into feed-forward or feedback-driven columns. So for example, here, when you have a seed region in the cluster of voxels that might be LGN pulvinar on the most bottom left slice, you see that most of the visual stream seems to be red. They all receive feed-forward input from that seed region. Whereas when we move, for example, here somewhere between V1 and V2, you see that the calcarein sulcus seems to be blue, receiving feedback, whereas most of the higher order visual areas still receive feed-forward input. And we can move along here, along the geodesic distance, to something that might be V5, where you see that most of the early visual areas are blue, receiving feedback, whereas there are still these further anterior areas bilaterally that still receive feed-forward input. So we can kind of map out the cortical hierarchy, the functional cortical hierarchy, on a column-by-column basis without predefined ROIs. Another application of non-BOLD layer-dependent fMRI, trying to approach more cognitive neuroscience questions, is one study that has been conducted together with Emily Finn. She will talk about this on Wednesday. And also Martin Havlicek will talk about the same paradigm on Wednesday from a more simulation perspective. And there we're looking at dorsolateral prefrontal cortex, where we also have very specific functional microcircuits. For example, we have these different cells. We have the black ones, the Q cells. We have the green one, the delay cells, and response cells that eventually trigger the motor system. And all of these different cells are distributed across different cortical laminae. And Emily Finn found this task that should engage these different cells differently by showing participants these strings of letters and then cueing them to either memorize these letters in the order they saw them or mentally rearrange them according to an alphabetical order, which is a very hard task and really engages the superficial layers here. And then later in the task, we show them random selected letters and they need to do a response decision, which is believed to be mostly engaging for the deeper layers. And this very simplified microcircuit can really nicely explain all the variance in the data that we are getting, here zooming into a CBV-weighted EPI that has this inherent T1 contrast. And you can see that for the response condition, we mostly see activity following the deeper layers along the cortical ribbon here upon smoothing of 1 millimeter within layers, whereas the alphabetization condition really results in activation parallel to that in the superficial layers. Better visible maybe also looking at the group results of 16 participants in the time courses, where we see that in the superficial working memory layers in green, the activity really shoots up as soon as the participants start to do this alphabetization task, and when they found a solution, it goes back to baseline. Whereas in the red signal trace, you see that the deeper output layers, they don't really care much about this alphabetization. They basically stay at baseline and only shoot up as soon as the participants found the right response and need to press the button. And if you want to hear more about it, check out Emily's preprint too. Another more neuroscientific application for non-BOLD layer-dependent fMRI that is really fully underdeveloped now still might be these so-called naturalistic tasks, aka movie watching, which might be very beneficial as a test case for non-BOLD layer-dependent fMRI because they allow us to do any kind of connectivity analysis without this ambiguity of like physiological noise-driven fluctuation versus neural signal fluctuations. And in order to approach this new regime, we use the same movies that I also used for the 70HCP project, meaning that we have about 100 participants already where we know with medium-high resolution what to expect, which kind of signal traces across the different brain areas. And we use the same movies also for our high-resolution protocols with 0.8 millimeter slab data. And on the bottom right, you see how much the Vaser can help here. For example, for the default mode network, you see that there are in BOLD a lot of false positives, especially at locations where the big veins are. With Vaser together, we can try to pin it down to more the neural components. And the kind of questions that we might be able to address with this setup refer to the sources of individual differences. So for example, here I'm showing any network, like the task-positive network, and from the HCP data, we know that most people have very similar time courses watching these movies in the task-positive network. However, there are still time frames that are more stable compared to other time frames. For example, when people watch wave patterns, the entire brain seems to do the same thing, whereas when you show them kissing scenes, they react differently. And then we can zoom into these parts where they are more similar or more different and see where these differences are coming from. And in the bottom, in the layer profiles, you can see that most of these individual differences are coming from the superficial layers, not the feed-forward, middle, and deeper layers. Which I think makes a lot of sense because they all watch the same movie, right? They have the same input through the eyes. The retina activity must be the same. The early visual areas extract the same low-level features. That must be the same. Only somewhere along the hierarchy, these individual differences are coming in as feedback here in the upper layers. Other questions that we might be able to address with this setup refer to kind of the hubness or directionality of already described networks, such as the default mode network, where we can use the HCP data, average them, and just extract the signal time courses of the default mode network for watching these movies. Then we use these signal traces as our regressor, as our design matrix, while people watch the same movies for our high-resolution protocols. And you can see that we can nicely extract the default mode network with that and also look at the layer-dependent responses. And you see that different areas here have different like layer profiles. For example, IPC or PFC are much more feedback-driven. They have these huge like upper layer peak on the left side, close to CSF, whereas the PCC seems to be much more inverted U and maybe more feed-forward driven. And comparing these profiles might in the future allow us to address questions about the hubness of an area. For example, suggesting that there might be one area that is kind of the driving force here and just dragging the other areas passively along with it with feedback activity. And I'm not sure how I am with time. Do I have 1 more minute?
1 more minute.
Okay, then I'll skip that. I want questions too. I will not talk about predictive coding, sorry about that. I thank all my colleagues and I thank you for your attention. Yeah.
So we have time for 1 or 2 very brief questions.
Yes. All right. My slides are gone, but I can refer you to the paper that has been just published in Science Advances, I think, with my collaborator Yinghua Yu. And there we look at the sensory cortex, which is even worse than the visual cortex in terms of thickness. And there you expect a very similar feed-forward feedback connectivity, namely that most of like expectation of being touched is expected in the superficial layers, whereas actually being touched comes from input from thalamus, mostly in the middle layers. So in that task, we already knew to expect that, and we just tried to see if we can actually also measure it by giving participants specific sequences of being touched. So they are touched in a certain order, they learn this order, and then always need to predict which finger is going to be touched next. In some instances, we left out the touch, so there they only had the expectation there, the predictive coding or prediction error, how Yinghua calls it. Whereas for like a lot of other control conditions, participants could not predict which finger is going to be touched next, but there actually was touch happening. So with this way, we could kind of dissociate between the feed-forward and feedback activity.
Thank you very much, Renzo. I would like to mention that we are open to receiving questions via the interactive system. Of course, you can use the old-style questions as well. Our next speaker is Jon Polimeni, and he will be speaking about pre-processing and analysis strategies for laminar fMRI.
Great. All right, cool. Thank you for the introduction. And so while I'm waiting for my slides, I'd like to thank Amir for organizing such a great educational session today. I'm really happy to be here. Thank you again for inviting me. And so I've been asked to talk about pre-processing. There we go. I've been asked to talk about pre-processing and analysis strategies for laminar fMRI. And so why I talk on this topic? Well, as we've seen, there's been growing interest in laminar fMRI in humans. It's known from invasive animal studies that different cortical layers have distinct functional roles, and therefore, a laminar fMRI may be the key to understanding functional architecture and brain circuitry in humans. However, it's very challenging from a data analysis standpoint. We'll see that conventional preprocessing and analysis techniques really aren't appropriate for laminar fMRI. This is partly because we're really at the edge of being able to detect fMRI changes in layers with our current resolutions. Today, we can't resolve layers either functionally or anatomically with our current voxel sizes. And also, it's important that there's some major interpretational challenges when it comes to laminar fMRI. We've heard about this many times this morning. There's a lot of hemodynamic coupling and spreading across layers, and therefore it's very difficult to infer the underlying pattern of neural activity from the fMRI signals we measure across layers. And so with this brief introduction, I'd like to give an outline of this presentation. I'll start by talking about anatomical reference data for high-resolution fMRI and ask how can we delineate accurately the cortical boundaries in our fMRI data. Next, I'll talk a little bit about preprocessing for intracortical fMRI and ask how do we preserve the high spatial temporal resolution in our imaging data. Next, I'll talk about analyzing fMRI data across both cortical depths and cortical layers. These are kind of related concepts but different, and I'll define them both in a few minutes. And there I'll ask how do we localize activation to individual layers with our current voxel sizes. And finally, I think this talk wouldn't be complete without a little bit of a discussion of the cortical depth dependence of the hemodynamic response. There I'll ask how non-uniform is the hemodynamic response across layers. So first, I think I need to say a few words about the anatomical reference data, because again, in laminar fMRI, we can't resolve or we can't detect individual layers. We need to estimate the layers from the cortical boundaries, and therefore it's really important to get accurate segmentations of the cortical gray matter. And so obviously we've been moving to smaller voxel sizes, as we heard this morning, both in terms of our functional and anatomical data. We wanted to revisit the assumption that a 1 millimeter isotropic, say, MP range acquisition was sufficient for accurately delineating the cortical boundaries in our human data. So we sat down with Bruce Fischel and the FreeServer team to make new modifications to FreeServer to allow ReconAll to be run directly on sub-millimeter data. So here's some examples of this new Recon stream on some 750 micron isotropic MP range data acquired at 7 Tesla. So with this higher resolution, we wanted to ask whether or not there'd be any advantages to going to higher resolution in terms of the placement of the surfaces. So here we looked across 40 subjects acquired at 7 Tesla. We compared surfaces reconstructed from 750 micron isotropic data with surfaces reconstructed from our standard 1 millimeter data. What we saw based on inspection is that the surfaces reconstructed from the higher resolution data indeed were more accurate in terms of their placement. We also saw pretty consistently across subjects that in the surfaces reconstructed from the high resolution data, the cortex was mostly thinner in most of the cortical regions. However, there are a couple of regions in which we found that the cortical thickness estimates were thicker, including in areas like V1, S1, M1, this is also, V1, S1, and A1, this is also seen by Matt Glasser. These regions are more heavily myelinated, also very thin. So we're seeing that there are many advantages to using higher resolution data. We see that those advantages do vary across different regions. However, overall, we are seeing that we do see more accuracy in the surface reconstructions with these high resolution data with this new processing stream. So next, the question is, as we go to higher resolutions, of course, we're going to be sacrificing SNR. And in some cases, this push to higher resolution can be counterproductive. So just to demonstrate this, here's some 600 micron data acquired at 3 Tesla. We ran this through our new FreeServer Recon stream. The surfaces looked okay, but there are a few places where we really weren't happy with the accuracy. And at first, we weren't sure if this was due to the higher resolution of the data or due to the low SNR in these high resolution data. So to address this, we averaged together 6 of these 600 micron acquisitions to bring up the SNR, and there we were much more happy with the accuracy of the surface placement. So what this tells us is that, it seems that the errors we were seeing before were really due to lower SNR in these high resolution data. And so the point is, as many of you go to higher resolution anatomical acquisitions to get higher accuracy, it's really important to be mindful of losses of SNR. In some cases, it may be more advantageous to go into a larger voxel size in order to get sufficient SNR to get high surface reconstruction accuracy. So as we heard this morning, there's been a lot of advances towards improving the quality of our API data, mostly for fMRI. And so we're asking, is it possible that API has sufficient quality to be used for anatomical imaging as well? So here's an example of some of our high-resolution T1 mapping with an API readout at 7 Tesla. This is a 1 millimeter isotropic whole brain acquisition. Very simple pulse sequence. What we do is we play a non-selective inversion pulse. We read out all the slices, so each slice gets a different inversion time. We repeat this, playing a second non-selective inversion pulse, read out the slices a second time, but this time we permute the order. So during the second inversion recovery, every slice sees a new TI. Now, if we repeat this enough times, in this case we did 24 contrasts in about 3 minutes, each slice sampled 24 distinct inversion times with which we were able to fit T1 recovery to each EPI voxel in order to generate a quantitative T1 map with an EPI readout. With this quantitative T1 map, we can then synthesize T1 weighted images, such as this one with MP range-like contrast, which we can then pump through FreeServer to generate automatic cortical surface reconstructions and parcellations directly from 7T EPI data. So I want to point out that this general idea of using EPI as an anatomical reference has been seen by many different groups. We heard about this morning, both in the context of 2D EPI and 3D EPI. And just for fun, we wanted to see if we could play this trick at 3T as well. This is a 1 millimeter isotropic whole brain acquisition at 3 Tesla, again showing that we have enough SNR for this idea at 3T as well. This is our one image where we acquired about 7.5 minutes. I think we could do this faster. And so the question is now, why bother with this? Why bother generating surfaces directly from EPI data? Now, I wouldn't advocate using this for morphometric analyses. We know that the cortical thickness will be affected by distortions in the EPI data, but that's kind of the point. The idea here is that now that we have enough quality in the EPI data, we can generate anatomical reference data using the exact same EPI readout that we use for our functional data, so that the anatomical reference and the functional data are exactly distortion matched. This gets around, I think, one of the biggest problems with fMRI analysis is to get accurate alignment of the functional data to the anatomical reference. This becomes more and more important as we push to higher resolutions in order to achieve sub-millimeter accuracy. So I think this idea of generating cortical surface reconstructions from anatomical reference data that's exactly distortion matched to the fMRI data, I think is a good strategy. And now, so as David mentioned this morning, if we do want to remove the distortion from the EPI data directly, we need to estimate a B0 map, which as many of you know is very challenging to do. Also, most of the distortion in EPI is along the low bandwidth direction, which as you know is the phase encoding direction. But as we go to thinner and thinner slices and smaller voxel sizes, we're starting to see that the bandwidth in the slice select direction is also starting to become somewhat low. So we are seeing distortions along the slice select direction as well. So just to demonstrate this, we implemented a pulse sequence very similar to a sequence that many of you have used, which you reverse the polarity of the phase encoding readout. Here we've alternated the slice select gradient polarity just to see how much distortion there is along the slice select direction. So if we compare the images from a positive readout to a negative readout, positive, negative, you can see how much displacement there are of the voxels in the slice select direction. So what this means is that there is appreciable distortion in this direction. If you do try to estimate a B0 map to correct for the distortions in the EPI data, it's important to take these distortions into account, as well as the distortions along the phase encoding direction. But again, if we acquire an anatomical reference data that's exactly distortion matched to the fMRI, these distortions are not something that we need to be concerned with. So another idea is that, of course, if we have our B0 map, we can try to perform EPI and warping in order to remove the distortions from the functional data so that they align to the anatomical data. But of course, EPI and warping causes resolution losses due to interpolation. So here's a map showing the resolution losses intrinsic to the unwarping process. This is EPI data distortion corrected with standard field map-based correction. So you can see that the pattern of the spatial resolution losses in these data look very similar to the B0 map itself. And I didn't use the audience response system, but a fabulous no prize goes to any audience member who can explain why this spatial resolution loss looks so similar to the B0 map. So again, on warping, the image requires interpolation, which causes resolution losses. They're also going to be a function of the voxel size. Again, voxel shifting on the image grid requires resampling and therefore blurs the data. So this is something we need to be mindful of if we want to perform distortion correction on the EPI data. Again, if we acquire anatomical reference data that's exactly distortion matched to the fMRI, this is not a concern. And with that, I'd like to move on to talk a little bit about preprocessing for intercortical fMRI. I know there's many different sources of resolution loss that could affect the ultimate resolution in our data. I showed you before how B0 distortion correction can cause resolution losses. Also, gradient nonlinearities have significant distortions. These distortions are a function of the gradient coil. They're also a function of the head position within the gradient coil. So we need to be mindful of these when we're comparing data across coils, across sessions, or even when the head shifts within a session. So here's a map of the resolution loss with one particular body gradient coil. You can see that the pattern here is very different from the pattern I showed you in a previous slide, corresponding to the resolution losses that we see with B0 distortion correction. In this example, you see there's not much resolution loss at the top of the head near a motor cortex or somatosensory cortex. We're seeing much more resolution losses around the center of the brain, around V1 and maybe the posterior cingulate. So any form of interpolation is going to cause resolution losses with different spatial patterns. Here's a resolution loss attributed to a motion correction with a simple head nodding by 2 degrees. We see for a distinct pattern of resolution loss. A head shaking by 2 degrees gives a different pattern of resolution losses. The point here, again, is that the interpolation causes resolution losses that are spatially varying. Now, the conventional approach is to apply each of our transformations individually. So, for example, if we have a motion correction transformation, EPI distortion correction and registration, followed by a gradient nonlinearity correction, these 3 consecutive steps of resampling give rise to a high degree of blurring. However, because the patterns of blurring in each of these steps is somewhat different, the blurring pattern tends to be spatially uniform. Now, this is what we see with a bilinear interpolation, sort of low-order interpolation. We can mitigate this somewhat by going to a higher-order interpolant, such as spline interpolation, which still gives a high degree of blurring, which becomes less spatially uniform. So I think a better approach that's been advocated in the Human Connectome project in a paper by Matt Glasser that I was involved with a few years back kind of advocates composing the transformations in order to minimize the number of interpolation steps applied to the data. So in this case, if we compose the transformations and perform a single resampling, there's less blurring. However, it does become somewhat spatially non-uniform. However, it's really straightforward to estimate the pattern of resolution losses if you know the transformations. So the idea here, again, is that we need to be mindful of these spatial resolution losses because in laminar fMRI, we're really at the edge of being able to detect these signal changes across layers, and therefore this can be another source of bias. So that's all I want to say about preprocessing. Next, analyzing fMRI data across cortical depths and cortical layers. So the very first step in laminar fMRI analysis is to simply sample the fMRI signals across cortical depths. And so therefore, we need to be mindful of the different levels of intercortical vascular anatomy in the cortex. Here are the vessels shown in the cast of macaque V1. Of course, we have these large PL veins that drain deoxygenated blood from large cortical veins lying along the PL surface. We also have these ascending venules that penetrate perpendicularly into the cortex. These are especially important in the context of laminar fMRI because they can be a source of a coupling of the fMRI signals across layers. And I'll be talking about that a little bit more in a few slides. And of course, within the parenchyma of this randomly oriented capillary bed. So we have to be mindful of the fact that fMRI signals across layers sample from different levels of the vascular hierarchy, and therefore we are expecting to see different couplings and different contributions of the vasculature as a function of cortical depth. So in order to perform this first step, we perform a cortical depth analysis. Here's an example of what I like to think of as a surface-based cortical depth analysis, in which we start by identifying the gray-white boundary. We reconstruct a surface there and evolve that mesh out to the PL surface, saving intermediate surfaces at various cortical depths. In this case, I looked at 9 intermediate or intercortical surfaces. I'm showing the cross-sections of these surfaces overlaid on the MP-range on the left there. So the idea behind this analysis is to simply identify our fMRI voxels to intersect each of these intercortical surfaces. So in the surface-based cortical depth analysis, we generate one cortical surface model per depth, and these surfaces in this example are placed at a set of equally spaced relative depths. Here I'm showing the 10%, 50%, to 90% depth relative to the total cortical thickness. So this analysis enables a cortical depth analysis over large expanses of the folded cortex. So just to demonstrate, this analysis technique, we wanted to look at the cortical depth dependence of spatial accuracy, in this case in gradient echo bold. So here we designed a visual stimulus that would give us a resolution pattern, a known pattern of neural activation in the primary visual cortex in the shape of this letter M, just for fun. We sampled it across depths, and here are the activation maps that we could see with this cortical depth dependent analysis. So we saw that the spatial pattern of the activation of the letter M was pretty good as we sampled near to the white matter, which is away from those large PL vessels. But as we progressively sampled closer and closer to the PL surface, we saw that the spatial fidelity of this pattern broke down systematically, but I think in an interesting way. Here's a fun artifact here. It looks like there might be a PL vessel that's cutting off the top corner of this M. So as Amir had said, I think the point spread function is a very useful tool to summarize the resolution in an acquisition, but as he also said, in a given spot, the resolution doesn't seem to be well characterized by a zero main point spread function. That's partly because our voxels are becoming so small, they're really sensitive to the local details of the vascular anatomy. So we can also use this approach to look how signal changes vary across cortical depths. We saw that in this study that the percent signal change near the white matter was about 1.5, whereas the percent signal changes that we measured near to the PL surface, which again are reflecting deoxygenation changes throughout the entire cortex, were about twice that, about 3%. So we see the steady increase in the sensitivity of the signal as we sample closer to the PL surface. We also looked at the spatial variability of the percent signal change, and we saw was that near to the white matter, we saw pretty much the same kind of low, small percent signal changes everywhere. But when we sampled near to the PL surface, we saw both big percent signal changes and small percent signal changes, which we think are reflecting the spatial heterogeneity of vessel sizes on the surface. At the PL surface, we have both big vessels and small vessels. So you have capillaries at the PL surface, and these big vessels are somewhat sparse. So we think that this broad distribution of vessel sizes may be the cause of the spatial variability that I'm showing here on the curve, on the red curve on the right. So what this is telling us is that these bold cortical depth profiles vary a lot over space. They vary very different from one location to the next, and the variability is most pronounced when sampling near to the PL surface, for gradient echo bold at least. These also show us that the profiles reflect different vascular contributions to the bold signal across cortical depths. So one issue that we need to be mindful of when using cortical surface-based depth analysis is the irregularity of the surface meshes. Both the vertex density varies both with the depth and curvature. So as I mentioned, we tend to start with a surface from the white matter. We generate a cortical surface reconstruction, propagate that mesh out to the PL surface. This propagation can lend itself to seeing a bunching of the vertices. This is shown recently in a nice paper by Kendrick Kay, in which he showed that with the free surfer method, there can be a bunching, a higher density of vertices, for example, in sulci at the PL surface. Now, this bunching can cause, when the fMRI data are projected onto the meshes, can be the cause of sampling biases. So there's a couple of ways in which we can correct for this. We can perform a vertex density correction. We can also use a more computationally intensive technique, which is really severely upsample the surfaces. But there's another approach. This is recent work from Valentin Kemper, in which he also looked at the regularity of the distances between neighboring vertices on the surface, and how they can vary across the folds. So the solution proposed and implemented in FreeSurfer is to replace this triangular mesh with a lattice or grid, or what I'd like to call a square quadrilateral mesh, where the density is uniform. So I think we'll be hearing more about that in the next presentation. So while these surface-based cortical depth analyses are really useful for visualizing the columnar patterns across the cortex and also across depths, there are some problems when we use these techniques for generating cortical laminar profiles. So the idea here is that if we consider our voxel grid superimposed on top of the folded cortex, we can see that any given voxel might be projected onto multiple surfaces, and this can be the cause of spreading of the signal across depths. So we can simply do away with these intercortical surfaces, calculate each voxel centroid in 3D, and then calculate the distance of that voxel centroid to the boundary surfaces of the cortex in order to assign to each voxel a cortical depth. So here's the differences that we see between so-called surface-based cortical depth profiling versus the proposed voxel-based cortical depth profiling. This is a fun example in which we're looking at physiological noise across depths. So in this plot here, just draw your attention to the blue curve. This corresponds to the percentage of variance explained in resting state BOLD with the retro-I-core regressor, which contains contributions from both cardiac and respiratory noise. We can see that the amount of physiological noise just very slowly increases in the surface-based cortical depth profile, very slowly increases as we sample closer and closer to the PL surface. However, if we consider the same data using a voxel-based cortical depth profile, we can see that the curve becomes much more L-shaped because there's less smearing along the depth dimension. We can better see that there's a transition between regions in which the voxels are sort of more dominated by noise from the white matter versus voxels being more dominated by noise from the CSF. So the idea here is that while there are some advantages to the surface-based cortical depth analysis, surface-based cortical depth profiling can smear activation across depths, and therefore to preserve resolution, I think it makes a lot of sense to use this voxel-based depth assignment. Now, we're doing something kind of strange in laminar fMRI. Many of you may be thinking that we're looking at these cortical depth profiles across cortex that's 2 to 3 millimeters thick. And typically, our voxels are about 1 millimeter in size, or maybe barely sub-millimeter. So how do we do this? We do this by generating depth profiles by pooling voxels across an ROI. So this is nicely demonstrated by Peter Koopmans. So what Peter did was he looked at some profiles across cortical depths, in this case, just in an example MP range. So first, he looked at the intensities across this green contour in the MP range, and he sampled it to, I'm not sure how many, like 100 different depths. And what he saw was that if he looked at the intensities across this profile that was oriented parallel to the voxel grid, he saw that there's about 6 or 7 unique intensities. Okay, and then he considered the profile along this, this red contour shown on the bottom there, and he also sampled the MP range across the depths, and he saw that, again, there's about, in this case, where the profile was oblique to the voxel grid, he saw that there's about 6 or 7 or maybe 8 unique intensities. But you can see that the transitions between these unique intensities occur at different cortical depths across these two profiles. So you can see where this is going. If he pools across many, many different profiles, in this case, he averaged the profiles across 25 different locations, you can see that this gives a much sort of better approximation to the smooth intensity variation across the cortex. Now, I like to think of this as being somewhat akin to what we do often in event-related fMRI, when we're trying to estimate the hemodynamic response function at a higher temporal resolution than our temporal sampling interval. We do this by jittering the onset times of the stimuli and acquiring many, many trials in order to resolve this at higher resolution. It's kind of akin to what we're doing here in laminar depth profiling. Now, this assumes that all voxels within the ROI have a similar intensity or fMRI activation across layers. This is a pretty strong assumption, and the natural inclination with this assumption is to assume an ROI that's pretty much as small as possible. But there are some problems when we do this. This can introduce sampling biases across depths. So if you consider a very small ROI in the visual cortex, it might be hard to see. There it is there. And we look at all the voxels sampled within this ROI, we can see in this histogram that not all depths are sampled equally. In this example, we see a lot more voxels are sampling from the peel surface, and also in this example, there seem to be a lot of voxels sampling from the white matter surface. What that means is that any profile that we generate across this ROI is likely to be biased by the signal contributions from these two locations. So what can we do? We can make the ROI a little bit bigger. In this case, if we look at the histogram again, it looks to be a little bit more uniform, still not perfect. If we go to a larger ROI, which takes up about half of V1, we see that the distribution becomes much more flat, indicating we have minimal sampling bias across depths. So this is a trade-off in defining ROIs for laminar profiling. On the one hand, we want the ROIs to be as small as possible, so the assumption that all of the voxels at any given cortical depth have the same activation within the ROI. We also want the ROI to be large enough to avoid these depth sampling bias effects. Now, it's one challenge in estimating the positions of layers across the cortical depths is that layer positions and thicknesses vary systematically with the folding pattern. Here we can see that in gyri, the lower layers are expanded. In sulci, the upper layers are expanded. So if we consider layer 4, this granular layer shown in green, what this means is that layer 4 is sort of pulled up towards the peel surface in gyri and kind of pulled down towards the white matter surface in sulci. I like to think of this as sort of exaggerating the curvature of layer 4. So this has been seen classically from both von Economo and also from Bach, who went one step further to try to propose a mechanical principle that kind of explained this phenomenon that could potentially be used to estimate the layer positions for macroscopically observable characteristics such as the cortical curvature. And so the Leipzig group really went and implemented this equivolume principle to try to predict layer positions from the cortical curvature. So they started off with a classic Laplace solution and then tried to estimate the position in this case of layer 4 and compared the predicted location of layer 4 to the detected location that they could see in their high resolution ex vivo data. We can see that the Laplace solution kind of minimizes the curvature of layer 4. As I said in the data, we see that the curvature is sort of exaggerated in layer 4. And it looks like the Laplace solution doesn't do such a good job in predicting the location of layer 4. They then looked at the sort of so-called equidistant approach, which assumes that layer 4 is at a fixed relative cortical depth across the folds. This did a better job. It didn't quite capture layer 4 perfectly. Again, this doesn't take into account this effect that I showed you in the previous slide. It doesn't vary with the folding pattern geometry. And finally, of course, they predicted the location of layer 4 using the equivolume principle. What they found was that this anatomically informed or inspired equivolume estimation performed best. And in fact, it's this approach that's used today in the majority of laminar fMRI studies for estimating the positions of the cortical layers. Now, this group went on in a subsequent study to note that we expect that the accuracy of the equivolume approach is expected to be greatest for small voxels. As we go to larger and larger voxels, the difference between the equivolume approach and equidistant approach may be minimal. So here's a comparison of the equidistant depth estimation and the equivolume depth estimation, animation provided to me by Renzo. Thanks, Renzo. So here you can see toggling back and forth between the two. As expected, the difference between these two depth estimations are more pronounced in places with high curvature, and they're pretty much the same in regions where the cortex is locally flat. So here's another example from Renzo looking at functional layer profiles using both the equivolume depth estimation and the equidistant depth estimation, showing that using the same kind of sub-millimeter CPV data that he mentioned in the previous slide. You can see in this case that the cortical depth profiles from these two depth estimations seem to be pretty much the same. And again, so what this shows us is that even with today's voxel sizes, the difference between the equivolume approach and equidistant approach doesn't seem to be very pronounced in our data. However, we do expect that as we go to smaller and smaller voxel sizes, we do expect the equivolume approach to perform better. There's other issues, complications in terms of estimating the positions of the layers. We know that there can be discontinuities in layers between adjacent cortical areas. The layer positions really jump around a lot across the cortex. You can see even between V1 and V2, we can see in the cell density in the myelin. So the mapping between cortical depth and cortical layer can vary a lot between neighboring cortical areas. So if you were performing a cortical laminar analysis in one cortical region, this might not be so important. But when you're trying to compare activation across multiple cortical areas, this is an important effect to take into account. Here's another example of this, showing varying thickness of the cortical layers across areas. Here I've picked a couple of primary sensory and motor regions. Of course, we know there's a lot of differences across the cortex. For example, there's no layer 4 and M1. We see that the layers really shift around quite a lot across regions, even between adjacent regions such as V1 and V2. Note also here that the thickest layers are around 500 microns. So what that means is that in order to comfortably resolve activation within an individual layer, we would need a voxel size of about 200 microns or so. So what this means is that in laminar fMRI, we very rarely, if ever, resolve the layers. So if I normalize these by the total cortical thickness, again, we can see how much the kind of relative contribution of individual layers varies across the brain, we can see how huge layer 4 is, relatively speaking, in V1 compared to even V2. So this can also be a cause of sampling bias in fMRI. We're going to get a lot more signal from layer 4 when we're looking in V1, and therefore we need to take this into account in our analysis. So another approach to kind of estimate the positions of layers is a nice approach proposed by Peter Koopmans, in which he sort of aligned the cortical profiles directly to the anatomical data. So if he looked at the original profiles in the baseline fMRI data, he saw that there was no distinct characteristics across depth. He also saw there's a lot of variability of the cortical profiles across the ROI. So what he did was he sort of slid the profiles around a little bit and adjusted the cortical depths in order to make the profiles more consistent across the ROI. And in this case, he saw this nice little decrease in the baseline fMRI signal around the location of the striae of Gennari. And this allowed them to see this increased activation within layer 4 in response to visual stimuli. So I really like this approach. It adapts the layer positioning to a detectable laminar feature, doesn't require an estimation or prediction of the layer positions. Now we need to be careful to not apply this approach directly to the fMRI data itself. This would be a little bit circular, but I really like the idea of aligning the depth profiles to some sort of detectable anatomical feature within the data. So we can also perform what we called anatomically informed smoothing. With this framework of small isotropic voxels and a family of intercortical surfaces, we can sort of embrace the full three-dimensional structure of the cortex. We don't have to approximate it as a 2D sheet, and we can perform kind of an anatomically restricted smoothing. And of course, we can position these intercortical surfaces however you like, based on the equidistant or equivolume estimation. So for example, we can perform smoothing within cortical layers if we assume that the activation is constant within the cortical layers. We can also smooth across cortical columns if we have a prior that the activation will be columnar. We can also perform more general, what we call intercortical smoothing, again, with this 3D smoothing kernel that tracks the folds of the cortex, or what we call a restricted kernel, which sort of avoids contamination from the white matter and noise contamination from the CSF. I just want to point out that this tool is now a part of FreeSurfer, so I encourage you to give it a try. So here are some examples of performing this intercortical or interlaminar smoothing. Again, a very strong prior on the data in order to smooth within cortical depths. Again, data from Renzo. Here we can see in the original activation, you can kind of see that there's like 2 peaks in the fMRI data across the cortical depths. This becomes much more salient after he performed this kind of laminar specific smoothing. And here's another example again where you can really see that the laminar specific smoothing really kind of pulls out this feature in the data. But I like this image because it brings up a very important point, and that is when we're smoothing, we're kind of imposing a prior on the data. It's really important to make sure that if you ever do smooth, to look at the data both before and after smoothing, to make sure that the smoothing doesn't introduce any features into the data, and that it's really just a way to visualize a pattern that you can see in the raw data. Okay, so last I'd like to talk about cortical depth dependence of the hemodynamic response function. And as I've said, in order to interpret the fMRI signals, we need to take into account how the vasculature and the hemodynamics vary across the cortical depths. It's been shown that the BOLD response amplitude and shape vary systematically across cortical depths. This is because of the spatial gradient of microvascular dilation delays. So this is nice work from Anna Devore's group looking at mouse cortex. You can see in this red ROI corresponding to the red trace shown on the right, the BOLD response is a little bit larger. Also note that it's got a very pronounced post-stimulus undershoot, and it also appears to have a little bit of initial dip in this red contour. If you compare that to the black contour, which samples from the parenchyma, there we see basically no post-stimulus undershoot, no dip, smaller response. So both the amplitudes and shape vary systematically. What this means is that any fMRI analysis using the same sort of canonical hemodynamic response function across all depths will also likely introduce addiction bias. You're more likely to see activation in the layer that has the hemodynamic response that matches your model. Now, this has been seen in humans as well, varying HRF features across cortical depths. This is work from Jon Polimeni from the Utrecht group. Again, you could see very similar characteristics to what Anna showed in the mouse, including a pronounced post-stimulus undershoot and potentially an initial dip from signals measured near to the surface, and these features are absent from voxel sampling from the parenchyma. So again, depending on which layer you're looking at, fMRI analysis using one single hemodynamic response function will introduce some detection bias. And lastly, so even if there is local blood flow control that is layer specific, we still need to contend with the known downstream hemodynamic effects, especially in BOLD fMRI. The idea is that deoxygenated blood sort of drains upwards through the cortex on its way out of the brain. And what that means is that BOLD responses in upper layers are going to reflect an integrated response from all the lower layers. So if you consider, for example, if there's neural activity in layer 6, we'd expect to see some local BOLD activation in layer 6. But as the blood drains upwards towards the surface, we're also going to see activation in layer 4. This becomes even more complicated if we have activation also in the top of layer 4, neuronal activation. We're going to see some accumulation of the BOLD effects in layers 2, 3. So what you can see is that not only is it an accumulation of the BOLD signal across cortical depths, but it's going to be really hard to infer which cortical layers there's the neural activation just by looking at the pattern of BOLD signal across depths. So the Donders group, David, tried to model these so-called laminar spread functions in order to account for where we'd expect to see BOLD signal if there's neural activation in any given layer. You can see that these modeled laminar spread functions are highly asymmetric, which accounts for this accumulation of the BOLD signal. Now note that here I'm really considering these downstream hemodynamic effects as they pertain to BOLD fMRI. But because of the interconnected intracortical vasculature, we'd expect to see similar couplings, maybe more complex couplings of activation in non-BOLD fMRI signals as well. Okay, so just to give some conclusions and final thoughts, I've shown you how new preprocessing approaches can avoid inadvertent spatial smoothing and respect intracortical anatomy. I've shown you there's a lot of complexities in the mapping between cortical depth and cortical layer that needs to be taken into account in these analyses because layer positions shift across the folding pattern and even dramatically across cortical areas, even adjacent cortical areas. I've shown you how the hemodynamic response varies across cortical depth, which is another source of detection bias, and is coupled asymmetrically between the layers. I think that I will say that I think there's more need for tools for this analysis that are integrated into common software packages. I think many people who do laminar fMRI kind of have a lot of home-written tools that they use for themselves. I think that we need to have more sharing of these tools and some standardization. And so final thought, and that is, in principle, laminar fMRI is easy, right? All we have to do is sample the signal across depths and see what we find. But I think that the challenges in laminar fMRI is the interpretation. The differences in the vascular responses across layers, plus this coupling that I mentioned, makes the interpretation really challenging. It's really hard to infer the underlying pattern of neural activity from these laminar fMRI signals. But I do think that if we can address these challenges, I think that laminar fMRI has a lot of potential for giving us sort of new insights, new fundamental insights into human brain circuitry and function. With that, I'd just like to thank a few people for contributing materials, especially Anna, Olivia, Villa, Shaheen, Natalia. Of course, thanks Renzo for the cool animations. Thank our funding sources for support, and thank you all for your attention.
Thank you, John. We have time for a couple of questions.
Yes. Yep. Yeah, that's a great question. So I think the simple answer is to map the HRFs across depths and use sort of a depth-specific or layer-specific hemodynamic response function. Yeah, so that's addressing the point that I made about how the shapes and amplitudes differ across layers. Yes, that's sort of one approach. Now, in terms of addressing the coupling of the responses across layers, that's a little bit more difficult. There have been some models, some sort of extensions to the Bloom model that have tried to take into account the coupling of the hemodynamic responses across layers. These results, while encouraging, are still kind of in the early stages. And so therefore, I think this is still an open problem, how to uncouple or decouple the fMRI responses across layers in order to understand the patterns of neural activity. And as I said, while we sort of have a sense for how these signals are coupled in BOLD or gradient echo BOLD fMRI, we're still learning about how these signals are coupled in non-BOLD fMRI acquisition. So I think this is still kind of an open question. I think we have seen a lot of success in laminar fMRI even without this. As I pointed out, there are many sources of bias we need to be mindful of. That isn't to say that these analyses aren't possible, but just something to watch out for.
So I would like to ask a question, if possible.
Yes, please, please.
One of the first slides, you showed a very nice detection of the cortical surface with multi-echo MPRH. So multi-echo MPRH gives you nice quantitative MRI of T1, but is it also advantageous in order to sort of draw the cortical ribbon to get cortical thickness or very nice, as opposed to MP2 range or just T1 weighted MP range?
That's a great question. And so it's a long answer. I'll try to be quick. So first of all, those are multi-echo, so those are not MP2 range, right? So I think we just did 2 echoes, so not really quantitative. I mean, you could try to fit T2 star from 2 echoes, but I wouldn't advise it. So these are really kind of qualitative images, just T1 weighted. What we have seen, though, with the MP2 range acquisition, which has a lot of advantages in terms of bias correction, we have seen that the MP2 range acquisition does cause some biases in the placement of the surfaces, partly due to the fact that the image reconstruction is nonlinear.